good. When Elizabeth asked me to speak at this thing on vector-borne plant viruses, I was kind of confused because I don't work on plant viruses. So I'm actually really excited that we've added this new topic here on comparison between animal and plant diseases, because I'm going to be giving you a talk about some of the work that we've done. I decided to at least talk on some of our vector disease work. This one is about Lyme disease in California. And you don't really think of Lyme disease as a big problem in California, and I'll explain some of the reasons why shortly. But this is work that was really spearheaded by my former grad student, Andrea Sway, who's now on the faculty at San Francisco State University, and a couple of people who've worked extensively on Lyme disease in the western United States, Bob Lane and Rick Otzfeld, in the eastern United States, who take very, very, very different approaches to the study of Lyme disease. And Andrea sort of refereed the whole project. And so the goals, I wonder, looks right here. Sure doesn't look right there. Let's see how bad this is, because it's not important here. But wait, now it no longer looks right. How bizarre. Okay, so the goals of our project are, we really have sort of four main goals. The first, which you can't actually see, we'll see if this is going to be continuing. I can mime out the whole talk. The first goal is to try to explain why we're seeing such different prevalence, different dynamics of this particular pathogen in the eastern United States versus the western United States. The second goal was to look at how the system responds to a number of different types of disturbances in the vertebrate community. And in particular, we really sort of started this project because sudden oak death, it is a plant disease. Sudden oak death was killing off oak trees, in the oak woodlands in Northern California, and we were wondering, okay, this change to the oak woodland composition is likely to have sort of knock-on effects on the rest of the system, on the vertebrates, on the ticks, and that could actually be affecting another disease in humans, so Lyme disease. I'm not going to talk too much about that part of the project, but we also went in and were a disturbance ourselves by going in and altering the host community and looked at the response to that. We've been developing models to help understand and predict how altering the vertebrate community affects Lyme disease risk in California. And as I was sitting in Lou's talk, I was wondering, okay, now which types of models am I doing this? And well, you can help me decide afterwards. And also, I'll be highlighting some of the main considerations of modeling this particular system, and we can wonder about how it would relate to vector-borne plant models. So that looks okay. So the system it involves a bacteria, the spirochete bacteria, Borrelia burgdorferi. So that's the pathogen in the system. It's vectored by ticks, and in the West Coast, it's vectored by Ixodes pacificus, the western black-legged tick. And then there are a range of different vertebrate hosts. There's some hosts that mainly feed the larval and nymphal stages of the ticks. Some hosts that are hosts mainly for the adult stage of the tick. And then humans are really just accidental dead-end hosts. Humans don't really play any real role in this. Occasionally we get bit by ticks. We get sick, but we aren't really transmitting it on and sort of affecting the dynamics in the real system. Probably don't need to say too much about Lyme disease to you all, because you probably have been to Lyme disease talks before. But it was first noticed in Lyme, Connecticut in 1975. There's a range of different symptoms, ranging from fatigue and chills and fever and so on, down to long-term neurological problems. And usually what we're interested in understanding is the risk to humans, which actually has a number of components, including what's probably really the most important risk is the density of infected nymphal ticks. We often also model the infection prevalence of nymphal ticks because it's actually a lot easier to model. And a big component that I'm not modeling at all is sort of the human aspect, that what humans do is really going to influence their own risk, how much time they come in contact with ticks. So One of the key features of tick-borne diseases, which it'll be interesting to see if this has any relationship to plant diseases, or the vectors that vector plant diseases, is that ticks have this life cycle that totally sort of drives the dynamics. And in particular, the ticks have to feed exactly once per stage. And so each tick is feeding exactly 3 times in its life. And this life cycle can take up to 3 years. So they're feeding about once a year before moving on. So usually the adult stage produces eggs that are uninfected. So there's not really effective transmission from the adult to the offspring. The larval stage, so the larval ticks, they're uninfected because they've come out as uninfected eggs. They feed once before becoming nymphal ticks. And so these nymphal ticks have a chance of becoming infected in the earlier stage. They have to feed again before entering the adult stage. And then they feed again, and those offspring come off uninfected. So the humans are really at risk mostly just during this feeding, this nymphal feeding during the nymphal stage, in part because these nymphal ticks are really pretty small and people don't notice that they've been bit by them. People usually notice when they've been bit by an adult stage, and it takes a while for the vector to actually spread the pathogen. So usually people can detect that. But these nymphal ticks are really, really teeny, and they've had one chance during this larval stage to pick up the infection. So it works out that the host for the tick during this larval feeding determines the nymphal infection prevalence. So Lyme disease, as I mentioned already, it's a huge problem in the Northeast. It's an increasing problem in the Midwest. And California actually does have a number of cases, but the prevalence is really pretty low. Although we actually have all of the players there. We have hosts that are good reservoirs for the pathogen. What's going on? I didn't touch that. We have hosts that are good reservoirs for the pathogen. We have ticks that can vector it. This is Ixodes pacificus out here, Ixodes scapularis in the east. And one of our questions is, why is it so different out here? There are a number of reasons. The first is because of the different tick species. The tick species has a different timing of its life cycle in the east versus in the west. And the timing of the life cycle in the East Coast is nicely timed to sort of optimize the transmission of this pathogen from one generation of mammals to the next. In particular, so it takes 2 years, has a two-year life cycle in the east, and the nymphs each year emerge before the larval stage. So the nymphs, which can already be infected, emerge. They can spread the pathogen to the young, vulnerable rodents out there. And later, when the larvae emerge in the summer, they can pick up the pathogen. Whereas in California, all of the action is really happening during the short window of time when there's water, if there's ever water again, where when it starts raining, everything comes out. And so everything's happening simultaneously. So there isn't really a nice effective way for the nymphs to infect the new rodents that come out and the larvae to pick up the pathogen later. So this difference of the timing of the life cycle is actually a huge contributing factor. Another difference is that we have a different host community in California versus the east. The white-footed mouse is the main player in the east, whereas the main good reservoir host in the west is the dusky-footed wood rat. And in the west, as in most Lyme disease systems, there's actually quite a bit of range in how competent the different species of vertebrates are at vectoring the pathogen. By that I mean of actually getting infected and spreading the Borrelia spirochete to the ticks. So in our system in California, dusky-footed wood rats are the most competent of the hosts. They are the thing that's probably maintaining the pathogen in the system. And mice in our system end up not being that important. They don't actually feed that many ticks. And we have this really important, totally incompetent host, which lizards, western fence lizards, not only do ticks that feed off of a lizard come off, not only do they sort of feed a large fraction of the ticks out there, but any tick that feeds on the lizard comes off uninfected. So an infected tick can actually get cleansed by feeding on the lizards. And it's part of the lizard immune system, the complement proteins that when they suck up the lizard blood, they get cleansed. So we have this kind of anti-host in the system, which is way cool. And so Lyme disease is really a pretty complex system. There isn't really just one spirochete bacteria. There's a whole diversity of them. We have a whole bunch of different vertebrates. There's the sort of stage-structured tick. And so one of the questions that I'm interested in is, okay, what is the appropriate level of complexity to model this system? And, okay, why do we want to model the system? Usually we want to model the system to be able to predict the nymphal infection prevalence and the density of infected nymphs and understand how changing any of the components will change those two risk factors to humans. And so when you think about the system, you wonder, okay, should we actually be trying to model the dynamics of the vertebrate hosts? So do we actually need to write an equation down for the dynamics of the lizards and each of those? And I think that's probably not. We probably don't want to include that because Lyme disease is really not the main thing that's regulating those populations. It's probably not even having that big of an impact on the populations of the hosts. And so in all the models that I've worked on, we've kind of just treated the vertebrate host as something out there. It might be changing, but it's not really something that we want to completely model. Instead, we might just want to be able to model the fraction of hosts in each of those species that are infected. And do we also want to model the dynamics of the ticks in each stage? And for the Lyme disease system, I think that it's all about the dynamics of the tick. Sort of a model of Lyme disease is really a model of the population dynamics of the tick, and the fraction of ticks that are infected in each of the stages. And so in trying to model these two components of risk, the nymphal infection prevalence and the density of infected nymphs, it turns out because of the kind of cool life cycle of the tick, models of nymphal infection prevalence are really easy, and they're really just sort of a bookkeeping exercise in that if you can figure out what the ticks are feeding on in this stage, and if you can figure out which host they're feeding on, how good of each of those hosts are at spreading the pathogen, you can actually figure out, so if they can figure out what they're feeding on here, you can figure out what fraction of them will be infected during the nymphal stage. And so writing down a model of nymphal infection prevalence is just like bookkeeping. It's just summing up over all of the different hosts the number of larval ticks that become infected by feeding on that host, divided by summing up over all the hosts the number of larval ticks that are actually feeding on anything. And so all of the different parameters in this type of sort of static model of nymphal infection prevalence, where because we have a sort of a year lag between each of these things, it's what the larval ticks are feeding on in 1 year, determine the nymphal infection prevalence in the following year. All of the different components of this are actually things that we can go out and measure in the field. Things like the population density of the different species, the body burden or number of ticks per host, the molting success of those ticks, the probability that an infected host of a given species will transmit the spirochete, and the fraction of those hosts of a given species that are infected. And so we did this. We went to a couple of areas north of San Francisco, China Camp State Park and Marin Municipal Water District, and set up a bunch of the 14 1-hectare plots, 7 at each of these locations, so that we had 1,400 sampling stations. They were also across a gradient of sudden oak death impact, which I'm not going to talk about that much today, and measured all those things to parameterize the model. So and trapped small mammals, lizards, drugged for ticks, and quantified the infection rate, the tick burden. Would you please stop that? And from that, figured out for the host that the larval ticks were feeding on in a given year, tried to figure out what the nymphal infection prevalence was the following year. And so this is actually a model where if we have the model right, it has to work, doesn't it? What we found is first just showing the infection prevalence in California. In the California system, really, there's just two key small mammal hosts involved, deer mice and wood rats. And the deer mice have about a 13% infection prevalence, and the wood rats have about an 18% infection prevalence, which is either really small if you have ever worked on Lyme disease in the East, where most of the rodents are infected, or it's really large if you think about it in California, where you think there really isn't that much of a problem. And if you look at all of the nymphal ticks combined, about 7% of them are infected. So if you go and plug everything into our nymphal infection prevalence model, and here are the data for a couple of the years, from year 0, we'll figure out what the prevalence would be in year 1, and for year 1, what the prevalence would be in year 2. A couple of things to point out: that if you look at the body burden for wood rats, if you pick up your average wood rat in California, it has 4 or 5 or 6 ticks. The average mouse in California has just a couple of ticks. But lizards have 20 or 30, sometimes 50 ticks on them. These lizards are just jam-packed with ticks. And that is one of the reasons that the nymphal infection prevalence is so low. If we look at the predictions of our model, we're actually, from our model, for some reason, underestimating at the true prevalence. This is if we look at that sort of site scale. I look at China Camp State Park in a given year, or Marin Municipal Water District in a given year. Our model suggests that we're really, from our model, we are really underestimating the true prevalence in a number of cases. And you might ask why. We don't know for sure, but there are a couple of possibilities. We could be missing some important host that is vectoring a lot of the ticks out there. Or another problem with the model is that we're just sort of looking at the means. We're looking at the mean body burden, the mean probability of a given species transmitting the pathogen. I actually think that we should be instead including a lot of the variability, and that we could be getting much higher prevalence because some of the individuals are actually acting as super spreaders in the system. So that's looking at the predictions at sort of the site scale, at the Marin Municipal Water District or the China Camp. We can actually also parameterize and test that model at the individual plot scale. So we have the data for all the bits of the system at that individual plot scale. For example, looking at observed nymphal infection prevalence in a given year, or the density of infected nymphs. And at that individual plot scale, the model doesn't do very well at all. This is looking at, for the different years, each of these points is now one of those individual plots, looking at predicted nymphal infection prevalence versus observed, and it's sort of scattering all over there. And part of the reason that it probably doesn't do a very good job at this scale is that maybe we're not looking at the appropriate scale to model. Maybe this really is too small of a scale to model it. And in particular, if you look at the size of our plots versus about the sort of average home range for any of the players in the system, it's probably not a bad scale if you're looking at at lizards, which have a very restricted territory size. And so modeling what's happening at a 1 hectare plot is probably okay for them. The mice, they have a bit larger of a home range size, and so we're probably not doing too badly there. But as we go up to the wood rats, which can roam for a much larger distance, or the deer, which is at an even much greater scale, maybe trying to model at this small spatial scale isn't the best thing to do. But we can use this model and make predictions from the model about the nymphal infection prevalence in California, and particularly how changing the relative abundance of any of the species in the system would affect the predicted nymphal infection prevalence. And the most important thing, so in our system it's really kind of interesting. The mice are really not having much of an effect at all, whereas in the yeast that's the main player. This is all as predicted, that if you increase the relative abundance of the wood rats, that increases the nymphal infection prevalence. But the big player is really the lizards. And if you reduce the density of lizards in an area, reduce the density of lizards this way, you expect the nymphal infection prevalence to go up. And so we tried this. We tried going into the system and actually reducing the lizards. We went to the same plots, and in 1 year after we'd sampled it for a few years, we went in and removed all of the lizards from half of the plots and followed the nymphal infection prevalence. And originally I was kind of worried that, okay, we're reducing the lizards, we're going to lead to Lyme disease outbreaks in Marin County, and we're going to be in big trouble. Basically, you can actually think of it could possibly go a number of different ways. Remove the lizards, and our really simple static model suggests that we should be getting an increase in the prevalence. But it really depends on what happens to the ticks that are no longer feeding on the lizards. So if those ticks, so it depends on sort of the foraging behavior of those ticks. If the ticks are able to switch over to another host in the places where the lizards had been removed, then we'd expect that when I say the density of questing larval ticks in that given year, those are the ticks that are still out there searching for a host that they haven't found a place yet. We would expect no increase if these ticks can just go over and find a new host. We would expect an increase in the larval burdens on the other hosts in the system. We'd expect, if those ticks are all finding a new host, we'd expect no change in the density of nymphs. We'd expect the nymphal infection prevalence to increase and the density of infected nymphs to increase. Alternatively, if there's no host switching, so we have this sort of stranded tick hypothesis, that sounds good, we would be expecting a slightly different pattern where the ticks aren't finding a host immediately, so they're out there still questing. We might find no effect on the larval burdens of other hosts, and we'd expect the density of nymphs to decrease, nymphal infection prevalence again to increase, and depending on the relative strengths of these two things, the density of the infected nymphs could go either way. What we actually found was this, where we found an increase in the density of questing nymphs. It looked like most of the ticks weren't actually finding a new host, although some of them were, and we did see an increase in the body burdens on some of the other ticks. And we found this decrease in the density of nymphs the following year. But the sort of surprising thing out of the whole thing was that all of our both of the sort of models predicted that we would see an increase in the nymphal infection prevalence. And actually, we weren't able to detect any real effect on the nymphal infection prevalence, which was really kind of a surprise to us, and we were still kind of left wondering how that happened. And what we ended up seeing was actually a decrease in the density of infected nymphs because we had a decrease in the density of nymphs as a whole. And so we actually didn't lead to an outbreak of Lyme disease in Marin County. We led to a slight decrease, but we still have this sort of questions that I'm not going to actually answer here of why we didn't see a change in the nymphal infection prevalence. That was the result that just seemed like it had to happen. But going back to this sort of questions of how we model Lyme disease in California, and if we now want to no longer just concentrate on the nymphal infection prevalence, but really talk about the density of infected nymphs, which is the real important component, this now is really just a model of tick population dynamics. And going to our life cycle and writing down this as sort of a model of the tick population dynamics, sort of this exercise of writing down this sort of model makes you realize that there's a whole bunch of unanswered questions about the population dynamics of ticks. In particular, questions such as how should we include the seasonality in the system? And one of the most important ones is where do we include density dependence in the tick population dynamics? What is actually regulating the tick populations? Is it the density of hosts? Are they actually limited by sort of tick space on the host, or how is that working? What do we actually assume about this tick foraging behavior, and what do we assume about the host infection dynamics? In particular, how are we actually getting infection persisting in the system from one generation to the next? I went to, you're not supposed to be able to read this, so I went to the literature and sort of summarized the models of tick population dynamics that I could find. And the models out there actually do a lot of these things in a whole bunch of different ranges, bunches of different ways. So this final column lists the type of model that was used, and they range from everything from ordinary differential equations to delayed differential equations to discrete time models with different types of time steps. But there doesn't seem to be, even in the models or in talking to tick experts, sort of a consensus on some of the simple things, okay, not that simple, some of the big questions in tick population dynamics, such as what's regulating tick populations? Where in the life cycle do you actually get density dependence? So we put together a model and made some assumptions about these various things. And our model ended up being basically, because we have this yearly pulse of all of the stages, it ended up being a discrete time model describing the dynamics of the larval ticks, nymphal ticks, and adult ticks, both in the uninfected and the infected class, and it ended up really just being a discrete time model. We decided to include the density dependence in the sort of the tick finding of hosts, and if there's too many, we basically assume that the hosts fill up, and if the hosts are actually full, which probably isn't the best assumption, that any extra ticks are lost, and so that ended up being a strong source of density dependence. But the big question of how we include the sort of tick foraging behavior, we just made an assumption about the ticks randomly coming in contact with the host, which I'm once again not sure it is the most realistic assumption. What the model does predict is as you vary the lizard density, for example, as you, for example, increase the lizard density, you get a decrease in the nymphal infection prevalence. It's not actually what we did find when we went the other way, but once again, model's not exactly matching up with the field. But the lizards in our system are feeding a huge fraction of those nymphal ticks. And so lizard density, increasing the lizard density or decreasing the lizard density can either increase or decrease the density of infected nymphs, depending on which of these two processes win out. And so I think I'm going to stop there. I want to thank Andrea, who really did all of the work sampling those 1,400 sites, plus Bob and Rick. So, any questions?